in order to accelerate medical and bio innovation, bring it faster to the market, and promote the bio ecosystem. I would like to take this opportunity and thank JLM-BIoCity team members, and especially Ms. Liana Naim, Ms. Mikey Horowitz, and of course, Mr. Joe Van Warren, President and co-founder of JLM-BIoCity, for the work towards this evening. And now, yes. And now to the part of the evening we are all here for. I'm honored to invite 4 key persons in the field of investment in the Israeli Life Science Review. Ms. Raisa Hakohan, Managing Partner at ARC Impact. Raisa, are you here?
Yes, I'm here. Thank you for inviting me. Lovely to be part of this event.
Thank you, Raisa. Dr. Ilan Goldstein, President at Merhavia Holdings and Investment. And I would like to say that we are honored to have you as an advisory board member of JLM-BIoCity. And also Mr. David Warburg, Managing Partner at Israel Investment Fund Group, and Dr. Maury Blumenfeld, Partner and Chairman at Our Crowd MedTech Venture. So thank you all panel members for being with us today. I will start by asking you all to present yourself, your organization, and its focus of investment. Mr. Cohen, could you kindly be the first?
I'm sorry, what was that?
If you could be the first one to present yourself and your organization, please.
Sure.
Thank you.
My name is Raisa Cohen. I'm the managing partner at ARC Impact. We do early stage investments in sectors of impact. So we've done assistive tech, ed tech, med tech, digital health, and silver tech. We write small checks fairly early, and we look for very promising high impact companies.
Thank you. Dr. Goldstein? Dr. Goldstein, can you please present yourself and your organization?
You're on mute, Ilan. You're on mute.
How about now? You hear me?
Yes, we do.
Perfect. My name is Ilan Goldstein, and I'm an angel investor in high tech in Israel. I got into the field about 10 years ago. Excuse me. And I do find it very exciting for the reasons that not only is it profitable, but also the investments have a big impact on people's lives and on society in general. Essentially, I invest in 4 sectors: defense, biotech, medical, agritech, and renewable energy. I can summarize and say that my investments have to do with the following areas of endeavor: protecting lives through defense and security, saving lives through biotech and medical, and improving lives through agritech and renewable energy investments. It's gratifying to be involved in many special advances that will improve people's lives in many areas. Part of what I look for is a special relationship or a special edge in the companies I invest in. For example, my public company, Merhavia, when I mean to say that we're the controlling shareholders, which is traded on the TAS, is invested in a new test for prostate cancer coming out of the Cleveland Clinic. That relationship attracted me to invest in CDX, as I felt that the Cleveland Clinic is a very special partner to have in a medical startup. As another example, I invested in a company called Precise Bio, which prints human organs on 3D printers. And what attracted me there is a special relationship that the company has with the Wake Forest Institute of Regenerative Medicine and the world famous Dr. Tony Atala, who is a leader in growing organs, regenerating organs. I'm excited to be here today, and I want to thank Joe Van Warren and the team. Joe is the one who introduced me to a few of the companies in which I've successfully invested in to this day, and which are flourishing as we speak.
Many thanks, Elan. Thank you for this presentation that actually show us, the values behind and actually show that what are you aiming at. Dr. Wahlberg, would you like to be the next one to present? David?
I think I was muted. I
Yes,
think
yes.
I think all of my dulcet words, I think, went into the ether and nowhere. So let me just first thank JLM-BIoCity. This is my first time, as I think everyone can tell, participating with this group. And let me tell you a little bit about me and the organization that I'm involved with. We have the Israel Investment Fund Group is based in Los Angeles. There are two principals and a couple of others who work with us. My background is as a lawyer and an investor and I'll say a builder of companies. That's what my passion is. My background involves high tech and biotech primarily. The Israeli Investment Fund Group actually is misnamed up till now. We have 4 investments in Israel and a 5th on the way, 2 in medical devices, 1 a winery, and 1 a high-tech slash a hard asset, or actually a plumbing company, but a sophisticated plumbing combined with software for water conservation company. We are about to invest in a 5th investment, which is another sophisticated medical device company, but you can see that our approach is very eclectic. What is exciting for us is each of those investments has been an individual syndication, but we are starting now raising money on our first fund. So we expect to raise a small fund of about $20 million. And our focus is an eclectic, opportunistic look at Israeli companies and Israeli assets. And that will cross all barriers and all types of investment, whatever sort of grabs our interest and that we think is promising. I personally have a background in entertainment, software, and biotech, and I'm quickly learning some of the other sectors that the Israeli companies are involved with.
Many thanks, David. Dr. Blumenfeld, please.
My name is Maury Blumenfeld. Can you all hear me?
Yes, we do.
Okay, good. So I'm a venture partner at Our Crowd. Our Crowd, as you probably all know, is one of the largest, well, is the largest what I call investment vehicle in Israel and one of the largest in the world. We have over 250, now it's over almost 260 portfolio companies. We've invested over almost $2 billion in them. We have a whole bunch of registered investors. The concept here is, in essence, to democratize the ability to invest in private companies. Usually, if you want to invest in a private company, you have to be a partner within of some sort with a major investor in a venture fund. Here we have a platform, and I think probably most people are familiar with what we're doing. My personal background is that I spent 35 years at General Electric in imaging. My last few years was as head of GE here in Israel. And as I tell everybody, I retired in 2002. And since then, I've been a failure because I failed retirement. Thank you.
Thank you, Maury, and thank you all panelists. The next session will include 5 exceptional companies presentations that will be followed by feedback from our panelists. First, I would like to invite Mr. Vic Levitin, CEO and co-founder at Diphtheria AI. Vic, please.
Hi, everybody. Good evening. Thank you for having me. Can you hear me?
Sure.
Yes, we can.
Awesome. Great. So hi, I'm Vic Levitin. I'm the CEO at Diphtheria AI, and we fight mosquitoes with mosquitoes. So here's the story. In his recent book, the professor historian Tim Weingart estimates that mosquitoes have killed half of humans who ever lived on Earth. That amounts to about 52 billion people that were killed by mosquitoes. Now you may say that's history, and today we have modern technology, modern medicine. But fact is that nowadays in 2021, the mosquito is the deadliest animal on Earth, killing more humans than all other animals combined, including humans. And every day now, there are more than 1 million new cases of dengue, a disease that is being transmitted by mosquitoes. And beyond the disease, they're just a huge nuisance. And you may think that poor citizens of developing countries, but due to climate change, mosquitoes are spreading fast throughout the developing world, throughout the developed world as well. This includes the US and Europe. So nobody's protected. And the problem is that we don't really have effective solutions. And we don't have vaccines or treatments to the diseases, and mosquitoes are fast becoming immune or resistant to the pesticides that are, in any case, they are toxic to us in the environment. So we don't really have a solution to this huge and fast-growing problem. Now, humanity does have a very promising technology called the sterile insect technique. It relies on a beautiful fact of nature that with many of the flying pests, including mosquitoes, females only mate once in a lifetime. And so if you release large numbers of sterile males into the wild, they will then mate with the females, thus suppressing the wild population. By the way, males do not bite. It's highly likely that you've never seen a male mosquito. Your humans were of no interest to them. And the sterile insect technique, or SIT, it works beautifully. A recent trial in Fresno, California showed a 95% suppression to the wild population. And this poses the natural question, why do we still suffer from all the nuisance, the disease, the death that mosquitoes bring upon us? And the answer lies with the need to release males only. Again, males do not bite. And the existing technologies to sex sort mosquitoes do so in the adult stage. Now, at adulthood, mosquitoes are very fragile and they have a short lifespan of a few days, which makes shipping them impossible. And so today you need to build a mosquito production facility in your every release site. And this is why the sterile insect technique is so expensive and it's not widely used today. This is where we come into play. At Diptera.ai, we've been able to build a proprietary technology, a platform that allows sex sorting insects at the larval stage. At this stage, the mosquitoes are very robust and they have about 2 weeks prior to becoming adults, which means that we can produce them in a centralized facility. From there, we can ship them across the country. And this unlocks economies of scale for the sterile insect technique. So instead of having to build a mosquito production facility in every release site, we can now offer a subscription service, or a SaaS, if you will, SIT as a service, where customers subscribe to a service and start receiving sterile males ready for release. We've recently completed our seed round of $3 million, and we are now working on an upcoming pilot in March next year. Thank you.
Thank you, Vic. Any question of the panelists? Yes, David, I can see you have a question.
Yes, I wanted to ask, there are other methods of mosquito control, some of them better, some of them worse. My question is if you could talk a little bit about the cost differentiation between what your proposed solution and what is out there already, or what you see as the competition.
Sure. So I'll give you a few numbers. If we're talking about the public health market, the mosquito control districts, we're talking about a total budget of about roughly, say, $4 per capita per year. This is what countries like the US, Brazil, and even most of Africa are spending on mosquito control per capita. This includes everything, the pesticides, the personnel, vehicles, airplanes, and whatnot. Out of that, about 30% goes to pesticides. So let's call it $1 per capita per year for pesticides. Now, with existing technologies SIT, or the sterile insect technique, costs right now about $10 per capita per year, which is 10 times more than what authorities are currently paying for pesticides. That's on the public health front. Now, talking about the B2C, the consumer market, it's simply impossible today because existing technologies, they require you to build a mosquito production facility in your backyard. This is not going to happen. Our technology actually unlocks the possibility to get to you as a homeowner. You will receive a container, like a weekly shipment of containers filled with sterile male mosquitoes. You just pop the hood and release them, and they do the work. And we are able to drive the cost down to be competitive both on the with the public sector and with the private sector.
Are you familiar with that? There's a company that sounds very similar to you called Forest Innovations. Are you familiar?
Of course, yes. I love Nitzan. We've actually met yesterday.
Oh,
yes.
okay. You're doing the same thing, aren't you?
There are a few companies now, but there's a renaissance around the sterile insect technique now. It's not a new technique. It's been developed in the 1950s. It's been widely used for the Mediterranean fruit flies in California, for example. But there's a renaissance around the SIT for mosquitoes now because cutting edge technologies like computing power and computer vision matured enough, and they're unlocking the possibility to really make breakthroughs in the space. Now, there are several approaches here. The approach that Forest Innovation took is they envision building these small factories near release sites. This is what they are doing in Brazil, and from these factories, you can actually protect a community. Now, this is great for some settings, but we believe that for wide adoption, a sterile insect technique as a service will be much more affordable.
Okay.
And to your question, they work with adult mosquitoes, not with the early stage mosquitoes.
Thank you. Any other questions from panelists?
How does this
Yes, Elan?
how does this tie in with the recent effort in Florida to release a huge amount of genetically modified mosquitoes? Can you say a few words about that?
Yeah, of course. I'll give you two answers to that. One is COVID-19 vaccines. Look at how many people are not happy to accept GMO into their bloodstream. That's one. And two is golden rice. We have a solution to world hunger. We have a solution to children dying of not having enough vitamin A. It's called golden rice. It's genetically engineered rice. The world is not ready to accept genetically engineered products. The problem is that in this case, when you introduce genetically modified organisms to nature, yes, your lab setting can show that it's safe, but when you release them in the wild, you're playing a long game with nature. You can't really predict what will happen. And Oxitec, who are doing the Florida Keys experiment, they had very, very alarming results in Brazil.
What were the alarming results? What happened?
Well, they said that their mosquitoes, they release a self-limiting gene into nature. So the gene should not be transmitted to the wild mosquitoes. Yet in the experiment in Brazil, they did find that there was some kind of spillage of the gene into the wild type. And to make a long story short, kind of mutant mosquitoes were generated, and nobody knows how immune are they to pesticides. So
wow.
Again, you're playing with nature. You can't really predict what will happen.
So they altered the germline of the genetic code of the mosquitoes.
This is what they do, yes. And again, from the scientific standpoint, this is definitely the solution. We are taking the not genetically modified way. We we use X-ray irradiation, which is the classical way to irradiate them, to sterilize the mosquitoes.
Ah, I see. Okay, thank you.
Okay, thank you, Vic. I would like to invite the second speaker, Ms. Hila Shaviv, CEO and founder of Gals Bio. Hila, please.
Hi, good evening, everyone. Am I presenting my screen or you are?
Yes, yes, you can share your screen.
Okay, I need
So you can control your
information.
screen.
Yes.
Mhm.
You can share now.
Yeah. Can you see? Can you all see my screen?
Yes.
Yes.
Yes, we can see.
Okay. Okay, hi, good evening, everyone. My name is Hila Shaviv. I'm the founder, the inventor, and CEO of Gals Bio. And I will present to you Tulipon. We like to call it the new internal wearable device for inside information. So all of us would love to know once in a while, how is our body doing? And mainly if we have any cancer growing inside of us, we would love to know or know about it at an early stage. If we are looking at women genital cancers, the only available detection methods are expensive and you can detect only at a late stage. At the same time, women experiencing on a regular basis all the time various kinds of vaginal discharge, including menstrual effluent and regular vaginal discharge that are gold mines of data to women's health. And this includes also cancer cells. Lately, there is accumulated literature connecting between the vaginal discharge and the cancers of various genital women cancers of women. And the goal of Gals Bio is to change the reality of these vaginal secretions currently simply being discarded and to give women the ability to monitor and screen their health from home with the accuracy of a lab. So this project is divided into 3 phases. In the first phase, we develop what we call the holy grail of vaginal secretion collection devices, where we develop a device which is used and look like a tampon prior to use, but it functions like a menstrual cup where it collects the vaginal secretion and it has one-way valve. So everything that is collected actually stays inside after the device is taken out from the body. Here you can see some of the results. This device has been tested as part of post-market study right here in Israel for the last year. And here are some of the results of our study. So far, we tested it with menstrual effluent. The next phase of this project is to actually take the menstrual effluent and other vaginal secretion and to research and to develop the technology for detecting biomarkers for cancers. And this is where we are at right now. In the future, our goal is to create an interaction with a smartphone where a woman could get identification direct to her smartphone with connection to the cloud. Our market size is obviously very big, and our goal is initially to focus on BRCA women around the world, which has gene mutation of BRCA, and their probability for various kinds of cancers is much higher, and some of them are even going through hysterectomies and surgeries in order to, because they don't have the technology to monitor their health and identify cancer at an early stage. We have various kinds of business models, and this includes B2B, B2B2C, and B2C. And also here you can see that our potential market size, again, is very huge. It's big. This is my wonderful team, and I wouldn't be talking to you if it weren't for them. It includes marketing people, engineers, doctors, and OBGYNs. And thank you.
Okay. So can I ask a question?
Wait, yes, just I'll stop sharing.
Hold it. It's going. Are you finished or?
Yes, I'm done. It just is, this is what backup. I'm just want to stop sharing. Yeah, that's it.
Okay, let me ask you, I didn't quite understand. You collect fluid that has cancer markers in it. Then what happens?
So this will be divided into two phases. In the first phase, it will be sent to a lab for analysis, and women will get diagnostic via an app. In the second phase, they will be able to take picture of the used device and to get identification straight to their smartphone.
So how much of this, I mean, have you already,
So
why am I supposed to believe you?
okay, so we already have, this is Tulipon. I don't know if you can see it.
Yes.
This is our Tulipon. This has been now clinically studied right here in Israel. And the fact that there is biomarkers for cancers within menstrual effluent and other vaginal secretion, this already exists in the literature. What I need to do currently is to find we actually already found, we are now negotiating our potential stakeholder collaborator that will perform as the lab to analyze the secretions.
So you're saying that on a smartphone, you will have a device that will be able to analyze, that's the part I didn't understand.
No, this, first of all, if we look at the short distance, within 3 years, we will be in the market and we will give women the ability to send their used device. This, the only thing will be on the smartphone is the results, okay? Okay, Asha, now in the future, what we'll have here, this will have a small window like a pregnancy test. She will take picture, or we can use other technology that I don't want to get into right now. But in any case, this will be, you know, I'm sure you know Healthy.io. You take pictures, and based on the colors, you can get identification. So we will have something similar to that. It's not the exact same technology, but it works the same way.
Thank you.
Thank you, Hila. Thank you. Any other question?
Yeah.
Do you have any, oh, hi, Iran. How are you?
Fine. Yeah, please, Ilan, be first.
Do you have any indications yet, Hila, of possible customers who are interested in using your test?
Well, I don't have anything signed yet, but I'm sure if I'll ask Angelina Jolie if she would prefer to cut off her breast or to have Tulipon, I'm sure she would be interested in having Tulipon rather than getting into menopause or doing breast implants at an early stage. So this is why we are focusing first on BRCA gene mutation women, which, by the way, I'm sure, I don't know if you know this, Jews are more, the percentage of the society, if in regular society is 1 to 400, in Jews is 1 to 40, the BRCA gene, okay? So no, I don't have anything stated or signed yet.
Okay.
Okay.
I have a follow-up question from Javier as well, like Ilan. Do you know or aware of any other companies in this domain that are looking on the vaginal discharge on biomarkers for cancer?
Yes. So where else? There is one in Israel. And there is a company called Gina Life. They are doing the same, but they are using a pad. Novelty, our major novelty is the fact that we actually developed a new kind of vaginal platform. To be honest, we started as a regular tampon to replace tampons. That's what my original vision was. After developing the device, we realized that this would be very hard to get into the market, so we pivoted. So this is kind of relatively new plan for us.
Right. So actually, Gina Life and MindUp, actually, I met them yesterday, and I really love the way that they have a swap, just like you have from Salavia. So you don't have to have really an internal, which females actually feel very comfortable to do as they used to do, but it's more easily. I agree with you just that the market is huge. The literature is correct. I mean, biomarkers like CA125 and others are not specific yet, unfortunately. And my kind of question is, are we by these biomarkers will actually, as medical doctors, suggest more surgery and not less? So I don't know yet. It has to be evaluated, and the clinical trials will not be easy. Nevertheless, I agree with you that the two other companies and you have a wonderful opportunity. That's I'm kind of giving you kind of a motivation to continue. Nevertheless, I would advise you to be very focused, as there are so many things you can have and do, and it'd be easy to have at least one claim and one kind of solution and extend it later, rather than to have a cup with things that you are looking for. So as there are other two companies, try to have a more precise what you would give, which they do not.
So I want to, if you don't mind, I would like to respond.
Actually, yeah. We'll respond very briefly, and well, you have received your feedback.
Yeah. So our biggest advantage that we are not using any absorbent material. So actually, I'm not losing any tissue. So I'm going to identify direct cancer cells within these fluids. I don't have to extract them from any absorbent material. And this is my major advantage. And since you are talking about more surgeries, this will be based on actual finding of cells. It's not going to be based on pH like Gina Life. This is going to be actual cells.
Okay,
thank you. Really, thank you. Just kind of in a comment, as short as we're short on time. When you're looking for cells are more directly from the same kind of organ, although when you go for a breast and you kind of collect from another kind of organ, you will prefer a biomarker than a cell because it will be more logic. So I like the idea, but try to see again the biomarkers and not only the cells. But thank you very much. I really enjoyed.
Thank you.
Thank you, Elad. Thank you, Eran. I would like to invite Dr. Seth Salpeter, co-founder and CEO at Immunic. Seth, please.
Great. So let me share my screen here. Everybody see the full screen?
Yes.
One second.
Okay. Yes.
Great. You can see the full screen or the presenter mode?
Full screen.
Okay.
Great. All right. So first of all, most important, I'd like to thank everybody for coming, specifically the organizers, Hamutal, Joe, and of course the investor panel. It's great to have these type of events. And I'll give you a short overview of Immunix and what we're doing here. So I'll start off by introducing you to the neutrophil, the star of our show. So the neutrophil, as many of you are familiar with, is the most populous of all the white blood cells. But paradoxically, it gets very little of the attention. So when we're talking about major therapies that are altering the immune system or immune cells, usually people talk about T cells, but the reality is that T cells, even though they're a very important cell, are a very small player, very small portion of the white blood cells. So neutrophils are really the most key in terms of establishing a healthy immune balance in the body and the major fighters against bacteria and fungi. And for many years now, the big pharma companies have known that in addition to their critical role in everyday homeostasis, maintenance of the regular white blood cell population, so they're critical in a large variety of diseases. So really, it runs the gamut of all the major chronic diseases, and I'll show you in the next slide, where neutrophils are actually coming into the specific organs that are involved in these diseases, the microenvironment, and altering in some type of negative way the effect of the tissue. So this includes the largest pharma companies, AstraZeneca, GSK, Merck, have now been working for something like 10 years to try and bring a neutrophil-based therapy to market. And the reason that they haven't really been successful is that because neutrophils are so important in the everyday healthy life of an organism, of a person, so when you ablate them and when you take them out and you stop them from doing their really important work, you end up causing something called neutropenia. Neutropenia, many of you are probably familiar with, is when you don't have enough neutrophils in the body, and as a result, you're susceptible to fatal infection from just the normal bacteria that's in your bloodstream. And when you look at the different types of diseases that are involved with neutrophils, so as I mentioned previously, the markets are huge. So neutrophils are really a key element, a key cause of the disease, a critical factor in almost all of the major diseases. So in COPD, in cancer, colitis, arthritis, the major immune diseases. So the negative effect of neutrophils in all of these diseases is well known. And the major drug companies have now been working for years to try and bring a neutrophil targeted therapy to market, but they've been stuck with this paradox where if you significantly alter and you destroy or you inhibit the population of the neutrophils in the body, so you're causing a lot of negative effects for the person, for their homeostasis. And that's where the Immunix technology comes in. So over the course of the last 7 years, based in the Hebrew University, Professor Tzvika Granot and Professor Tzvi Friedlander have been developing a new system to be able to target and manipulate neutrophils in a variety of diseases. So what they've developed, and you can see it here in this diagram, is basically what we call a neutrophil targeted nanopeptide, right? Nanoparticle, excuse me. So what you see either via oral or inhaled administration or IV administration, a patient is given this formulation of a nanoparticle, which is in the blue, and it's coated by a targeting peptide, a peptide that has been uniquely developed to bring its payload, the drugs inside of the nanoparticle, to neutrophils. And it does that by targeting a receptor, a protein that's on the neutrophil, that's called CD177. It's a receptor that's only expressed in the neutrophil. And when the nanoparticle is coated with this peptide, the nanoparticle brings the drug, it brings the targeted payload specifically and only to the neutrophil. So what you see here in 3 steps, the peptide is targeting to the neutrophil. As a result, the nanoparticle then joins inside, it emerges inside of the neutrophil and releases the payload. And the payload can be either a drug, it can be siRNA, and it can even be mRNA. So we've developed the platform so that we can use a variety of different nanoparticles in a platform, either PLGA or liposomes or LNPs, to be able to target a variety of different moieties to the cell. And this you may be familiar with a lot of the work that was done recently by Moderna and by Pfizer, where they are also using LNPs to target mRNA. So it's based on a lot of work that has recently become very popular in the clinic about targeting mRNA to specific types of cells. So as I mentioned before, the big advantage here is that we're able to come in and specifically and very uniquely do a surgical targeting to neutrophils without ablating them, right? So if you come and you block their differentiation, then you're completely taking away the ability of the neutrophils to stop bacteria or fungi in the body. But what we're coming to do is we've created a platform technology where we can specifically deliver a unique drug or gene sequence to alter a neutrophil in a specific type of diseases. And this will allow the neutrophil to continue living in the body, continue maintaining its ability to fight the bacteria. But we're coming and we're looking disease by disease to understand how the neutrophil is acting negatively and specifically manipulate that pathway. So for example, in COPD, which is our first indication, it's known that a specific activity called ROS secretion is something which is very detrimental in COPD. So our approach is to come and specifically alter ROS secretion from the neutrophil while maintaining the neutrophil alive and allowing it to continue fighting bacteria throughout the body. So in our first indication and our first drug development project, we're focusing on COPD. COPD is the 3rd leading cause of death worldwide. It's a huge market. Pharmaceutical companies have made big efforts in the past to develop neutrophil-targeted therapies for COPD. It's a very dynamic and well-known factor in the disease, but they failed. As I mentioned, they've even seen efficacy in the COPD in a variety of different trials, but again, they've seen neutropenia, the destruction of the neutrophil, which has been very dangerous. So they've discontinued the trials in that area. And our goal is to treat COPD specifically by modulating neutrophil function. I won't get into the details here, but you can just see some of our already existing preclinical proof of concept work. We have already shown in mouse models of COPD that we're able to use our neutrophil targeted nanoparticles to improve the preclinical markers, the mouse markers of COPD. And I'd be happy to get into that later with anyone who's interested. So just a quick snapshot of the things that we're working on when we're focusing on COPD. As I mentioned before, there are a variety of different diseases where neutrophils are critical. So two additional areas that we're working on is in the oncology area and in colitis. We actually have active discussions with pharmaceutical companies going on now about how we can partner with them, either with their existing molecules as payloads or on completely new development projects. And we're also interested in targeting specific mRNA or siRNA sequences to undergo neutrophil modulation. So the key behind all of this work is clearly the team. I introduced them very briefly before, but Professor Tzvika Granot and Professor Tzvi Friedlander are not only two very prominent professors at the Hebrew University, but they're actually the world experts in neutrophil biology, discovering some of the key paradigms in the field, N1 and N2 polarization and high and low density neutrophils. And they're very actively involved in the company. Also, what's extremely beneficial for us is having a chief medical officer. Tzvi is also the director of COPD here at Hadassah Medical Center. So the expert team is also supported by an advisory board of world experts in the fields of nanoparticles, including Professor Chazi Barinholtz here at the university, Professor Ronit Satchish, who is well known also for her work on nanoparticles. COPD work in the US is being led by the former director of COPD care at Mass General. And as well in the field of nanoparticle delivery, we have some world experts in the US and Dr. Dale Drummond. So I'll just wrap up with a brief summary. So neutrophil directed therapy is really a huge unmet need. There's almost no companies in the market now that have made great strides in terms of altering their ability, their function in diseases, either in small biotechs or in the large pharma. From our discussions with a lot of the pharmaceutical companies, we know that this is a major area of interest and they're very eager to partner with us, especially as we have already shown preclinical efficacy in a variety of different mouse models. We're looking to really drive this to the clinic as quickly as possible by partnering with some key players in the US, hoping to get our COPD into phase 1 clinical trials by early 2023. And happy to take questions.
Thanks, Seth. Raisa, would you like to ask Seth some questions? Any questions?
Yeah, Seth, you said that you came out of Hebrew U. Have you taken any funding to date? Can you walk us through your
Sure, yeah, no problem. So the initial development stages, or the mid to late stage development in the university was funded by a Kamin grant. So the project also received 3 full years of Kamin funding, the last year of which was partnered together with VLX. And then VLX, about half a year ago, came in and spun the company off of the university as part of the incubator program. So that's basically the runway for the funding up to date.
And are you currently raising?
Yeah, so we're looking to raise an additional sum to help us specifically expand beyond COPD. So the initial funding from the from VLX and also from the Kamin was focused on COPD specifically. We're looking for additional funds to expand the preclinical work in colitis as well as in oncology, and also to further some of the specific development work that we need to get to the clinic. So I'd be happy to discuss particular sums afterwards if you're interested.
I'm curious as to why you started with COPD. Can you speak to that a little bit?
Yeah, so it's a good question, and it was basically for 2 main reasons. So first of all the diseases, it's most clear biologically and physiologically that neutrophils are a key player in COPD, right? So if you stop any physician, any lung expert in COPD on the street and you say to him, are neutrophils important in COPD, he'll be like, yeah, we know the big pharma have been working on it for years. Every one of them has failed because of this problem with neutropenia. So it's low-hanging fruit. We know that if we can alter COPD we can alter neutrophils in COPD, then there's almost no doubt that we'll be able to achieve efficacy in the clinical trial. So that's number one. And number two is that in a lot of the other areas, there's a great deal of saturation, right? So if you come into there are tons and tons of companies with all sorts of different stories about oncology and colitis and immune manipulation. But COPD is somewhat of an open market at this point. There aren't so many really intense competitors in COPD, mostly because people have tried to make strides in it and they haven't really succeeded. So we think that coming with a really novel approach where it's clear that if we can successfully manipulate the neutrophils, which we know we can, so it's really an easy way to show in a first indication that we can show efficacy, right? And then once we have developed a lot of that in the first indication, then we can try more challenging indications like cancer, where the market is more saturated and the challenge is a little bit greater, but we've already proven ourselves. So I think that those are really the two key elements in terms of moving us to COPD. But we've taken this to the pharma companies, and they all say to us, we're very interested in COPD, but tell us what you're doing in cancer and colitis as well, right? So when we get this feedback from Pfizer and from GSK and from AstraZeneca, so we're really compelled to try and really move this forward as fast as we can now, to be able to show them, to further our preclinical results and show them what we have. So these are things that they're interested in actively partnering, but if you can't come to them with super early phase data. You have to have something that's really a little bit more, rounded out and complete. And that's really the goal of the additional funding.
Thank you, Ted. Thank you, Rita. We'll move forward to the next speaker now. I would like to invite the current CEO of PRC Biomedical, Mr. Arye Huber, please. Arye, are you here? Okay, so we'll maybe move forward to our next speaker. Evan, are you here?
Yes, I am.
Okay, so I would like to invite you, Evan, Mr. Evan Spiro, CEO at Vectored Photonics. Please, Evan.
Hi, everyone. Today, I want to share a big concept in AI smart building products. A big idea that starts with a small step in the health and wellness vertical, or not even a step, but a trip and a fall. My name is Evan Spiro, and our company Vectored has developed a robotic personal attendant. Imagine that you own a robotic attendant that can automatically serve you like an $80,000 butler would, but which starts at $199 per room. However, as consumers have lived for years without a personal aide, and they're not going to buy one unless there's a big need, we're starting off with a big problem. Next slide. Rooms are dangerous places. In the United States alone,
we don't see the slides.
Yeah.
No slides, Evan.
Mike, are you there with the slides?
Yeah,
Yes.
1 second. I'll show it to you.
I'm sorry, guys.
My apology.
I didn't
think-
miss too much,
No.
guys.
You had great momentum.
Got our attention.
PRC Biomedical, right?
No, I'm Vectored Photonics.
Vec-
The last guy,
Sorry, my
Vectored Photonics.
apologies.
Yeah, we skipped something in the middle.
Okay. Second. Okay, great. There we go.
Thank
You're
you.
on.
I'm on. All right, now we've got to get my view back again.
Sorry.
No problem. All right, so we were saying that we needed a problem in order to get this product into people's nursing homes and rooms, etc. And in the United States, a child is killed by a falling piece of furniture every 2 weeks. Someone has a stroke every 40 seconds. Globally, an estimated 684,000 individuals die from falls yearly. Have you ever asked yourself, why is it that we have a smoke detector in every room to protect our property from a fire? However, as of yet, there is no person in an emergency detector to alert us of a person in the room who's in danger. Maybe they're having a stroke, a heart attack. As the most dangerous room in the world, nursing homes. How many of you heard of the horrors that go on in nursing homes? Here is an example on this slide. James Dempsey, a decorated World War II veteran, begged for over an hour for help. When the nurses finally came and put on his oxygen mask, they discovered that the machine was broken. They laughed. James died. They were sentenced to prison, but that didn't return James to his loved one. So there has to be some kind of a solution. So as its first application, Vector is using its unique artificial intelligence room robot to detect if an elder is experiencing an adverse event. It then makes a call to a guardian or adult child who may then approve the call to 911. A call center and monthly charge is not needed as it is by our competitors. So what does the product look like? If you look up off to the left, you can see the minimum viable product, Garty. It looks like a typical wall lamp located in the patient's room. Off to the right is the $199 mature embedded eye attendant product. It's a typical light fixture installed in the ceiling, but now there's a computer inside. And this computer integrates on this bus, the room's many single-purpose devices, sensors, appliances. For example, we have in there a speaker, a microphone, and also a universal remote controller that you can control by your voice or gestures. So when an event occurs, the all-knowing machine learning robot uses its devices and appliances to serve recognized individuals in the room according to their learned personal preferences. No, it's easy to use. Services are automatically provided just the way you like it. There's no need to ask as there is with Alexa. So our target market, as we said again, we're starting B2B sales in nursing homes because we determined through market surveys a burning need on the part of the worried children of the elderly themselves, and the nursing home owners. They're, our AI system has been trained to identify neglect, abuse, and falls. The algorithms that we have determine the severity of our guess, how good it is. And if necessary, the processor automatically will make a video call to a guardian in 911. And this is what it looks like on the Guardian's screen. Off to the left, you can see that if you make a call into the app, you'll see the current biostats for wellness tracking of the individual who's being of interest. And then on the next screen, you can see the call that's made out by the system if a fall or suspected abuse has occurred, the Guardian gets to see. And if somebody even tries to tamper with the system, we know who that is. So how do we get this product out? It takes a multidisciplinary team, and we have guys who are wild about this product, making it happen, and we have the skills to make it happen. Team was put together around with the skills needed in order to make this happen. Addressable market. Next slide. The residents of skilled nursing facilities are about 1.8 million beds in the USA, 3.6 million in Europe. Okay, and we have a liability loss insurance that's paid per bed from on average of $2,300, and in our target state of Florida, $6,000 per bed that's paid by an operator. So there's an incentive to buy our product. There's market size is large, even in this niche market of 2.7 billion total addressable market sales. The future market, of course, the fastest growing population segment worldwide is the elderly. There are 2 million in over age 90 in Japan alone. Competition, there is competition. There's a lot of it. There's a product by Philips, which is the best-selling product in the United States for fall detection, an alert button, similar to what we have here at Yad Sarah. There's Alexa, Google Home. There's surveillance cameras, such as Google Nest. We put many, many functionalities together, and that's where we're different than the competition. Traction, we've been around operating 2016. Some of our patents have already been issued, some are still pending. We have received 3 consecutive R&D grants for $1.1 million, including investment from family and friends. We began prototype testing and we've obtained market validation. We have a letter of intent from the Dorot Geriatric Hospital, that's up by Netanya, for a check within their geriatric hospital. So we're seeking in a post-seed round, next slide, 1.2 million in continuing investment. And we're going to be completing our testing on what we've already developed. And then we're going to be introducing this into nursing homes. Within 1 year, we should begin sales to nursing homes. And that base will allow us to get the data that we need in order to go over to the mature product and a much bigger market, as we said. So I hope you're excited about this business opportunity as we are. The team at Vector is committed to seeing our person in an emergency detector in every room and every office as soon as possible. We're calling out to strategic partners, investors, and VCs to come join us on this avant-garde, high-impact mission. And considering the mature market size, it's also an incredible business opportunity. So being part of saving lives and lessening the suffering of millions worldwide interests you, please call me, Evan Spiro, to discuss it further. My contact info is off to the right. Thank you very much.
Thank you, Evan. Any question from the panelists to Evan?
I have a question.
Yes, Elan.
I'm curious to know what you think of a competitor named Sophie Hub, because I have invested with them in the past.
Say the name again of the
Sophie Hub out of Australia. Sophie Hub.
Sophie Hub. All righty. Not familiar with them. I know of many American and European firms. I'm not familiar with them.
I know the market is very crowded with a number of competitors. So what's your
what specifically do they do?
They have sensors that sit in the rooms of the nursing homes, detecting falls and making sure that the people are safe, etc. Similar to what you're trying to do.
Right. Well, we're trying to detect we're a smart home device. Okay. So first off, we turn on the lights and the air conditioning and everything else. So do they do that?
I'll have to research it and get back to you.
Okay. So I don't imagine that the breadth of the product that we're capable of doing, because of the unique electronic architecture that we have, is able to do what our system does. Okay, so we're not only falls, our AI has learned to detect abuse, neglect. A pill reminder will come out of this. We can also play Netflix. Okay, are they capable of playing Netflix? So, and we can play the Netflix into their hearing aid.
Netflix is the most important out of all the functions.
Could be, because how often do people forget to put on their pendant that's a fall detector? But our device is in use all the time.
Yeah, of course.
Well maintained for all the things that are around the house. So that's why we will be there to get your back the first time you fall. The other guys can't say that.
Thank you very much.
Thank
You're
you.
welcome.
Any additional question from panelists? Okay, so we can move forward. And I would like to invite, thank you, Ivan. I would like to invite Ayi Huber. I hope we can hear you now. The chair and CEO of PRC Biomedical. Arya?
If there is any problem, maybe I can replace him. I mean, I'm familiar with the project.
But yeah,
So-
but it's you know, it's required his agreement to that. So thanks, Ariel.
Arya Orbach and Arya Huber work together. So
Yeah.
I would very strongly recommend that Ariel Orbach replace him because it seems that the audio problem is not solved.
Okay, so Arya, we apologize, your audio problem. Yeah.
Okay, but you just you put on the presentation, or
We can put the presentation on,
Okay.
yes. And please speak briefly, but you're running out of time. Thank you.
okay. So what we're
Thanks, Ariel.
talking about is a unified kit for the different blood tests that are currently done in the blood bank, together with different characteristics of red blood cells that influence the functionality and viability of the red blood cells that affects the outcome of the blood transfusion, mainly the survivability of the red blood cells after transfusion. So just next slide, please. Next slide, please. Yeah, okay. So the goal is to have efficient blood storage management and matching the right blood unit to the specific patient, meaning that today the blood units are at the blood bank are stored in the blood bank for 42 days without any specification to the characteristics of the red blood cells derived from the different donors. What we do is we prioritize the blood units according to their actual characteristics and their ability to be functional. Next slide. Next slide. Yeah, okay. So today the current policy in the blood bank is the first in, first out, meaning that from the day of donation, the 42 days or 35, depending on the solution of the preservatives. And there is no test for the quality of the red blood cells. Today, when you come to donate, for example, if a woman comes to donate blood and her hemoglobin level is about 12, it's okay. And the man hemoglobin level is about 15 and it's okay, but they're on the same shelf. This is just a matter of simple way to demonstrate that, but we're looking on much more profound differences in the characteristics of the red blood cells that determine the functionality and survivability. According to the FDA, there should be 75% survivability of red blood cells within 24 hours after transfusion. However, about 1/3 of the cases do not meet these criteria, which of course leads afterwards to poor transfusion outcome, repetitive transfusion, and different side effects. Next slide. Just that you will understand more or less what we're talking about. So the characteristics of the different ways to characterize red blood cells, one of the most common one is the flexibility, their ability to go through the the small capillaries, because the diameter of the round red blood cell is about 8 microns, and it has to go through capillaries which are as narrow as 4 microns, which means that if they're not flexible, they can cause different fractions and also break into debris. So it's harden the all the system. Next slide. Okay, so this is just to show if the red blood cells is rigid and it can't be flexible, so it causes these infraction in the capillaries. If it happened in the hand, it's not so bad. If it happened in the brain, it might be more severe. So just next slide. Okay, so our solution is a kit that contain all the different antibodies for the different blood tests that are commonly done in the blood bank. On top of that, we have specific biomarkers for the characteristics of red blood cells that can determine their ability to survive, and also to determine for how long you can store them and they'll still be functional after blood transfusion. Next slide. So in practice, just that you take the blood sample from the blood unit, and also you take blood sample from the patient, you process it, and it takes about 2 hours. Just as a proportion, today the tests that are done for blood units in the blood bank takes about 8 hours. And we can determine all the different blood groups according to, including the minor antigens and the presence of pathogens. And of course, afterwards, to have an optimal blood transfusion. Next slide. We have actually now coming 3 different patents. One is already published, becoming national in different places, and the other one is in the PCT stage, is still not on the web, and the third one is just provisional at the moment for the specific biomarkers, and also for the specific kit that conclude all the different tests on one array. Next slide. So we have as I said, we have the different patents. We had different clinical trials. Now we're finalizing a clinical trial actually in Rambam Hospital for the survivability. We already established a very sophisticated lab in China that they're finishing finalizing the kit, and afterwards it's also this lab is capable of production, at least in the first batches, for a larger clinical trial, and also for a later stage for the market. Next slide. So of course the market is huge, because although today we give less blood units in terms of the regulation, that today the people who have more than 8, their hemoglobin count is 8 and above, don't get blood units, but still because 50% of the blood donations that are transfused are to the older population. Of course, it's growing, so the actual number of blood units is growing, and the market is huge. The main customer, of course, are the hospitals and the blood bank in the hospital, but there can be also a market in some other medical institutes. Next slide. So the team is Arya Uber is a CEO. I'm the CTO. We have Mr. Wang. He's a pharmacist who's working with our partners in China. Tom Mei is our medical scientist. Is a full PhD and MD, and we have advisory board. And that's it. So I can take some questions.
So many thanks, Arya, for this spontaneous placement. Make some noise. Mikey, can you mute?
Are there any questions?
Yes, please. Yeah. So we can hear. So okay, thank you, Mikey.
Can I just ask what?
Please.
the Rambam trial, that is the clinical trial, do you know what phase is that? How far into the clinic are you at this point?
Okay, so this is it's like, because it's a diagnostic, we don't have to go through all the stages of like a drug. So this is considered as phase 1-2, because there is no matter of safety, it's just to show the ability. And afterwards, there is just one more trial that we have to go through, because what we're doing now is we're just measuring or predicting the survivability, but we don't do it from the day of donation, meaning the full kit's supposed to predict on the day of donation the storage duration and also the survivability, and to prove that. Just because of logistic, we didn't have enough money for the whole clinical trial, so we break it into two. So we did already one in terms of the duration, and now we just show the survivability and our capability to show better survivability with our kids.
And let me ask a naive question. Who, if you prove this out, who would be your customer? And then who would you market it to or sell it to?
Yeah, so because it's the unit test include also the other component of blood banking tests. So basically the different, let's say here it's going to be either MADA or Tel Shomer, the center red blood, the center of or the Red Cross in the United States. So just the very center places, but also hospitals that have blood banks, because the blood banking is committed to run two tests. One, it's when you donate, and the other one is during storage. You have to run at least one more test. So basically, these are the customers. Just a very central customer, and also the different hospitals, but also some small hospitals like in Hadassah, they have their own blood bank with donors. So they will also have to use these kits.
Okay.
Can you hear me, by the way?
Now we can hear you.
You can hear me? Okay, can I add a little bit? So first, sorry for my first big computer didn't work and I was muted for some reason. Anyway, our customers, to your question, our customers will mainly be the hospital blood banks. There are about 50, 60,000 like that in the world, maybe more. And also central blood banks and other institutes. Of course, we intend to join in a partnership with one of the big companies in the world, one or more big company in the world that are dealing with the blood tests. And there will be some interest from them.
Many thanks. Thank you, Arya.
Sorry again about what happened.
No, you know, Ariel, you know.
Yeah, he did very well, and I thank him.
So thanks, Arya, and thank you, Ariel. Actually, we're running out of time, so I apologize, but we will have to run to the next part. And so thank you, Arya, again, and thanks, Ariel, and thanks all speakers for your impressive presentations. And I would like to say that we have definitely learned a lot about your promising products that are taken from different fields within the life science arena. And I would like to invite our panelists to provide their precious feedback, which I'm sure that both speakers and all of us listeners are waiting for. And the first feedback will be provided to Diptera AI. Well, David, you were the last one to question. Would you like to be the first one to provide your feedback?
Sure. I always enjoy this part when people that have not been studying and working and doing what people do in their company tell people what they're doing wrong and what they can do better, because we don't. It's a good life. This is my real diphtheria is it's a very impressive. It's an impressive concept. It's clear that you're far along in getting things ready and commercializing. Let me give you some what I hope is constructive criticism or focus. I think it's important that you focus on who certainly your initial customers would be. And I sort of didn't understand the answer that we think we can send mosquitoes to, to houses, to people living in the suburbs, and they will get a weekly or monthly allotment of sterile male mosquitoes and happily let those go in the backyard. I think from a narrative standpoint and in terms of raising money and advancing the company as quickly as possible, it would seem to me, and again, there may be a direct response to this that tells me I don't know what I'm talking about, but I would try to sell to people that already are comfortable with the need to eradicate the mosquitoes in a particular area and the people that have funding for it and a built-in market, and that would be the mosquito control districts and other public or private, the companies or the entities that are already tasked with getting rid of mosquitoes instead of creating a market, a new market for it. And that also would tie into the individuals living in a house are not concerned with dengue fever. They're concerned with getting bit. But the more serious an immediate problem would be, gee, if you could help eradicate dengue fever, malaria, and Zika, and so on, I think that should be the focus. But I have to say, I liked both the written presentation and the presentation you gave today. It's very positive.
Thank you, David. The second company to receive the panel's feedback is JLM-BIO. Could you provide your feedback, Raisa?
Of course, thank you so much. It's a very interesting concept. I did take a close look at GenoLife. I'm very interested in the biomarker space, as well as I think femtech is an interesting and growing market, especially in terms of fundraising and investment. So I would love to see where you guys go. I think hardware is a tough sell, especially in the femtech space. But I would love to see, as you guys continue to collect data and iterate, I'd love to see how you guys progress.
Okay, thank you, Raisa. Maury, would you like to provide your feedback to Vector Photonics?
To which one?
To Vector Photonics.
Well,
I'm
the guy with the robot.
Ivan Spiro.
Yes. Can you see me first? Hold on.
Yes, we can.
Okay.
See you.
Well, I thought it was very, very interesting, particularly as an old guy, who I think this is something that the future houses will definitely have to have. And from my point of view, getting in there as quickly as possible is very important.
Okay.
I liked it.
Thank you.
Okay. Thank you. Ilan, could you kindly provide your feedback to PRC Biomedical, sorry.
PRC.
Ilan,
you are still in mute.
thanks, Iran.
Sorry about that. I found it very, very interesting. I don't understand all the biology necessarily around red blood cells so easily, as far as how they the mechanics. But if they can improve the technology of how the blood is stored and how the transfusions take place, I think it's an amazing technology.
We can prove.
Merchavia, my public company, actually did invest in another company that has to do with medikits that can go in the field and do blood tests, like in Africa and other very rural areas. Maybe Yoel Ezra, who has these. It's called EFA, Engineering for All. Did you come across this company with a portable blood kits? I'm not sure if you came across them or not.
I heard about it, but it's different what we do. What we do.
Different, right? Exactly. But it sounds like amazing technology, and like you said, the market sounds tremendous, and you can market to, I think you said 60,000 blood banks. So it's really a vast potential. Very, very
hospital blood banks.
Sorry.
60,000 hospital blood banks.
Hospital blood banks. Wow, wow.
Another few thousands central blood bank.
That's unbelievable. Really tremendous. I have to look into it further, and it sounds very, very fascinating. Thank you.
Okay, thank you, Ilan. Well, David, let's get back to you. Could you please provide your feedback to Immunix?
Sure, for Immunix. Fascinating, interesting, obviously an important, if you can do what you think you can do, it will be a huge advance in the science and a huge advance for the community, and the targets you have are obviously, they're huge. I mean, if you could do anything with any of those, with COPD, with cancer, with whatever, obviously one doesn't have to worry about valuation of the markets. I think I just have two comments for you at this point. One is that when in doing the presentation, which was excellent, I would go into a little bit, I would have a slide on what it is that all of Big Pharma has been doing that has not worked, and that differentiates where you think you're going with your technology, and for both for a layperson and for a scientist that would know more of what you're talking about. I sort of put you in the category of people that are focused on Alzheimer's. A lot, billions of dollars literally have been spent trying to find a cure or treatment for Alzheimer's, to no avail. If you think you've got one, I think you should explain, even in just a few sentences, to a layperson as to why you think you've got something that others don't. And then along with that, just because you are so early stage and preclinical, which is always difficult, I like the idea that you've gotten funding up to now, as I understood it, from grant work, and I would focus on that for now. And I would not be talked into trying to build your platform for every possible indication, even if AstraZeneca or Merck is whispering in your ear saying, do that. They just want you to do their research. I would tell you to focus, focus, focus. I think your idea of getting one, let's start with COPD, prove the concept, and the platform can expand from there. That would be my advice, but really excellent presentation.
Many thanks, David. So thank you all. I have one more question that I would like to ask all panelists, and I would like to ask you to answer it in brief, please. The question is, well, if you had to choose, what would you give as your most important tip for entrepreneurs? Like one sentence advice for early stage companies from your own point of view. Mr. Goldstein, could you please be the first one to answer this important question?
Sure. I'm speaking as an angel investor, and I often get personally involved with strategy for the companies where I invest. I like to stick in my 2 cents, whether as an observer or a director on the board or in any other capacity. Often I connect companies together as a matchmaker, or shadchan, like we say in Hebrew. So I think when you pick an angel investor, at least as opposed to a VC, it's important to find someone who brings a lot of added value to the company, and someone who on a personality level is someone you can feel comfortable working with to develop the company.
Thank you.
Sure.
Important. Misa Cohen, Raisa, can you please share your opinion?
Sure. I hesitate because I think I'm the most green investor on the panel. My co-panelists have far more experience than I do. So I would defer to their advice. I would just say that when you're on the entrepreneurial side, it's very easy to get into sell mode. And when you're dealing with very, very early stage investing, I find a lot of it is just building up trust in the team and trust in the market. And so everything you do, every interaction, whether it's in a pitch or an email, is a data point for me. And all those things need to add up into belief in the team and belief in the company.
Thank you, Rita. Dr. Wahlberg, what do you think of this question?
I would say a couple of things that I've already touched on. One is to stay focused, and to not get excited about all of the possibilities of the thing you're working on, stay focused on what you're doing and the potential market, and some of the specifics is, and how would you enter that market? Who are you actually going to be talking to actually, when you have a commercialized product, who will you be speaking to make your sale?
Thank you. Dr. Blumenfeld, Maury, what would you consider as your?
I agree with, can you hear me?
Yes.
Yeah. Okay. I agree with everything that everybody said. David really put it in a very nice way. The only thing that I have noticed all the time is that there isn't enough, when a company gets to a certain level where they start to have to address their customers, they have to start to understand the difference between marketing and sales. And I know that when I first came to Israel, there was no understanding of that difference. When people said, when you said, well, marketing, so they'd always talk about sales. And so there has to be a good understanding of marketing, what it means, and how to get it to what will be eventually the customers.
Thank you. So thank you for this tips. We are going to the end of the event. I would like to thank all entrepreneurs and panelists for educating and impressive presentations and ideas which make us realize, every time, how every development step and every aspect should be calculated from the beginning, from the very beginning, considering all relevant factors, like you said, the difference between marketing and
sales.
And sales, yes. And I would like to thank each and every one of you for being here with us today, and invite you, of course, to connect to JLM-BIoCity LinkedIn group, and also take the opportunity right now and stay and participate in the breakout room, just to listen to interesting speeches and start connecting and create synergies. Thank you all.
So how do we get to the breakout rooms?
Mikey, would you like to explain, or should I explain that?
I'm just open the breakout rooms. People should be invited, or they should move automatically to the breakout rooms. Can you hear me?
Yes, we can.
But where do you find them?
Okay, great. So they're at the bottom. There should be 4 boxes that should say breakout rooms. If you can't see the 4 boxes, then click on more, and then you should see breakout rooms. You should be able to choose which breakout you want to go to. If anyone has specific things or is having problems, is welcome to stay here, and I'll move you to the next breakout room of your choice.
And
Just let
panelists,
me
you will be
know.
automatically affiliated to a specific breakout room, right?
I can see that they've already been moved. Yes.
Yes. Okay. Thank you.
Okay. If anyone wants to let me know, just let me know via voice, as in speak to me, because
I'm supposed to join David Wahlberg.
David Wahlberg. Okay. Let me go in. Allow me. Anyone else?
I would like to join Ilan, and also
Ilan?
to David.
Anyone else?
How do I join David Wahlberg? Eliakim?
Yeah, there we go. again, the rooms that are there are the rooms of the panelists that spoke.
Where are the links to the rooms?
Can you not see at the bottom? I think I sent you to a room, but I don't know if you did it. You want to go to David again? Sorry, I just, yeah, okay.
Mikey, I'm going to stop the recording, okay?
Okay, great. Thank you.